Okay, team, let's take a look at module 2.3, where we're talking all about plant transport. We're going to go through everything to do with transpiration and translocation, as well as measuring the rate of transpiration too. Make sure you give this video a like, make sure you subscribe as well so you don't miss any of the other videos that I've made breaking down all of the GCSE biology you need to know. Let's get into it. So the first thing we need to know when it comes to plant transport is the organization of the tissues in the leaf. So let's go through our layers initially. We're going to start at the top here with our waxy cuticle. Now, the waxy cuticle is a transparent layer that lets light pass through it, but it is impermeable to water. That means it doesn't let water pass through. This is really important because it stops the leaf drying out and reduces the amount of water loss. Plants that live in particularly dry conditions, they will have a thicker waxy cuticle to reduce the rate of water loss. Imagine something like a cactus. Sometimes they look a bit shiny on the outside. That's because of the thick waxy cuticle. Underneath the waxy cuticle, we have our first layer of cells, and this is the upper epidermis. Now, the upper epidermis, this is just a protective layer. The cells, they are transparent, so they're going to let the light pass through as well. Underneath the upper epidermis, we have the palisade layer, sometimes referred to as the palisade mesophyll. Mesophyll basically just means layer here, or tissue. So the palisade mesophyll, this is where the majority of the photosynthesis is taking place within the leaf. The leaf is an organ. It is made up of multiple different tissues, and the purpose of that organ is to photosynthesize. Now, the palisade mesophyll are adapted to carry out more photosynthesis. We can see in the diagram here, we have lots and lots of those little green dots, and that is because they are the chloroplasts. So the palisade cells have lots of chloroplasts. We can see they're also quite long and sort of column shaped, and that's to maximize the amount of light that kind of ends up in those cells, the amount of light that it captures. Underneath the palisade mesophyll, we have the spongy mesophyll, or the spongy layer. And the spongy mesophyll, we can see, has lots of gaps, lots of air spaces. This is going to massively increase the surface area, and that's going to increase the rate of diffusion of gases in and out of the cells. Now, some important gases here might be carbon dioxide diffusing into the cells for photosynthesis, or oxygen diffusing out of the cells that's been produced in photosynthesis, or perhaps at night, oxygen diffusing into the cells for respiration as well. Remember, respiration is also happening during the day, but the cells are producing oxygen in photosynthesis, so the oxygen is there ready for the respiration. The other big important gas that might be diffusing in and out of these cells is water vapor, and we're going to touch on water vapor more when we get onto transpiration. Also, within this spongy mesophyll layer, we might also find a vascular bundle, or a vein. Now, the vein is made up of the transport tissue, the xylem and the phloem. We're going to look at the xylem and the phloem in just a second. Underneath the spongy mesophyll, we have our lower epidermis. This is our protective layer on the bottom of the leaf. And within the lower epidermis, we have these little gaps. These are the stomata. So the stomata are the holes that allow gases to diffuse in and out of the leaf. Things like carbon dioxide need to diffuse in, things like water vapor need to diffuse out. And the opening and closing of the stomata can be controlled by the cells next to them. These are the guard cells. Now, the guard cells, they are going to change shape depending on the water availability. If there's lots of water available, that means water is going to move into the cells by osmosis. As the water moves into the cells by osmosis, they swell, they get bigger. And because of the shape of the cell wall of the guard cells, that means that the guard cells pull apart from each other, opening the stomata. Now, if there isn't very much water available, that means that the water is going to leave the guard cells. As it leaves the guard cells, they become flaccid and they lose their shape. And as they lose their shape, that means the stomata closes. So if there isn't very much water available, the stomata close. And if the stomata are closed, that is going to reduce the amount of water that is evaporating or diffusing out of the stomata, and therefore reducing water loss. We can see at the very bottom of the leaf, we also have a waxy cuticle here, but it is slightly thinner than the waxy cuticle at the top of the leaf as well. And that's just because the majority or the top of the leaf is going to be exposed to more sunlight, the top of the leaf is going to be hotter, and therefore we would see more evaporation from the top of the leaf. So we need a thicker waxy cuticle to prevent that water loss. All right, let's take a look at transpiration now. So transpiration, as a term, the definition we need to know. The definition is the movement of water up the leaf, or up the plant, sorry, up the plant, or we could say from the roots to the leaves via the xylem. So it is going up the plant. So how does transpiration take place? Well, firstly, it is a passive process. That means that the plant doesn't need to expend any energy in order to make this water move. The movement of the water out of the leaf is due to evaporation. And as the water evaporates, the surface tension of the water is going to pull on the water in the xylem. The xylem here forms a continuous column of water. It's a little bit like a straw. Imagine how when you have a straw in your drink, you suck on one end of the straw and that draws the water all the way up the straw. What that does is that pulls the water out of the roots. Now, as the water moves out of the roots, that's going to reduce the amount of water that is in the root hair cells. And as the amount of water reduces in the root hair cells, the cytoplasm becomes more concentrated. And so water is going to enter the roots via osmosis. And that is then going to replace the water that is lost from the leaf. As I said, transpiration is a passive process. Now, the evaporation of the water is due to the kinetic energy of those water particles. So the water particles have energy, but the plant doesn't need to expend any energy due to released from respiration in order to get that water to evaporate. It's happening just because of the temperature of the surroundings. So let's take a look at the different factors that affect transpiration. Well, one of those first factors is going to be the temperature. Now, the temperature of the surroundings is going to affect the evaporation because if we increase the temperature, we're going to increase the kinetic energy of the particles. And if we increase the kinetic energy of the particles, this is going to increase the evaporation. Another factor that's going to affect the rate of transpiration is going to be the wind speed. On a particularly windy day, we would see more transpiration. So an increase in wind speed. What this is going to do is this is going to remove the water vapor from the area surrounding the leaf. And if we remove the water vapor from the area of the surrounding the leaf, it's going to reduce the kind of water concentration in the air around the leaf, basically the humidity. This is going to decrease the humidity around the stomata. And if we reduce the humidity around the stomata, what this does is this increases the diffusion gradient or the concentration gradient. So this increases the concentration gradient. Another factor that's going to affect the rate of transpiration is going to be the humidity in the air. Now, humidity is the amount of water vapor in the air. If it's really, really humid, the air has lots and lots of water vapor. And that means that there's going to be less of a concentration gradient between the inside of the leaf and the air itself. And if there's less of a concentration gradient, that means we get less diffusion. And so we get less transpiration taking place. We get less of the evaporation of the water out of the leaf. So an increase in humidity means there's more water in the air, which means there is a decrease in the concentration gradient. And this is the one that people quite often get the wrong way around. The consequence of this is going to decrease transpiration. This is why plants that live in very dry conditions like deserts have to have special adaptations in order to reduce that rate of water loss. They might have fewer stomata perhaps. So, on that theme of number of stomata, let's talk about another factor that affects the rate of transpiration. This is one that students quite frequently get wrong. This is light intensity. Now, light intensity, as we know, is related to the rate of photosynthesis. The higher the light intensity, the higher the rate of photosynthesis. But photosynthesis also relies on other things, such as carbon dioxide. So, if there is a high light intensity, the leaf is going to open the stomata to allow more carbon dioxide in. So, as light intensity increases, more stomata open, and this is to allow carbon dioxide into the leaf. If more stomata are open, there's going to be more available space for water to evaporate out of the cell, out of the leaf. And if we get more evaporation, this is then going to lead to an increase in transpiration as well. Now, the mistake that students make when they think about light intensity is they say that increasing light intensity increases the rate of photosynthesis. Photosynthesis requires water, so more water moves through the xylem into the leaf. And unfortunately, that is incorrect, because around about 80 to 90% of the water that moves through the xylem evaporates out of the leaf. Photosynthesis contributes a very, very small proportion of the amount of water that is used by that leaf. So the reason why the light intensity affects the rate of transpiration is due to the number of stomata. Now, other things that are going to affect the rate of transpiration that are related to the number of stomata are also going to be things like the size of the plant and how that relates to the number of leaves, how the number of leaves relates to the number of stomata, and also the stomatal density, how many stomata there are per millimeter squared. If there are more stomata per millimeter squared, that means there's going to be a greater rate of transpiration. Next, let's talk about translocation. So translocation, our definition is going to be the movement of dissolved sugars around the plant through the phloem. Now, the dissolved sugars are made in the photosynthesizing parts of the plant, the leaf, and other parts of the plant need those dissolved sugars. The roots, for example, need the dissolved sugars for respiration, to release energy for active transport of ions, or for cell division perhaps. Or perhaps the plant is going to store those dissolved sugars as starch in the roots ready for the winter when the rate of photosynthesis is going to decrease because of reduced temperature and light intensity. So the plant needs to move the sugars around it. It's also going to be moving things like amino acids as well that are produced in the leaves. Now, the leaves we might refer to as the source because they are what is producing the dissolved sugars, and the roots we might refer to as the sink or the other parts of the plant. Now, translocation is an example of an active process. It is an active process because energy is required from respiration in order to load those dissolved sugars into the sieve tube elements, the parts of the phloem that the dissolved sugars move through. Remembering our adaptations of the phloem as well that we looked at in our module 1 video. If you haven't gone and checked that out, do go and take a look at it. The phloem also has the sieve tube plates, which are pores in the ends of the phloem vessels that allow the movement of the solutes to move freely between them. The phloem also has the companion cells, and the companion cells help to load the solutes into the phloem, into the sieve tube elements. And because they're doing lots of active transport, they need to have a high rate of respiration, which means they would have lots of mitochondria. Okay. So how do we measure the rate of transpiration then? Now, this is another one of the experimental setups you need to know about. What we're going to be doing is using a piece of apparatus called a potometer. Now, a potometer is set up like the diagram below. We have a piece of plant material, a cut shoot. What we're going to do is we're going to cut that shoot at an angle, and that's going to increase the surface area. And then we are going to have an airtight seal around the piece of the shoot. And that's so that no air gets into the xylem. Remember what I said about the xylem being a continuous column of water? If any air gets into that, well, it's not going to be able to function. The water's not going to move through the xylem. Imagine if there was a pocket of air in a straw, you wouldn't be able to suck up all the liquid. Now, that shoot is in this tube containing lots of water. And that tube is contained to a capillary tube. And in the capillary tube, we have a very small air bubble. And as the plant consumes that water and takes it in through the stem, the water is going to move in this direction. And so the air bubble will move in that direction. And the distance the air bubble moves, we can use to determine the rate of transpiration. Our rate of transpiration is going to be equal to the distance the bubble moves, or perhaps we could find the volume from that distance using the volume of a cylinder. So we would do the volume divided by the time. In order to reset the air bubble and move it back to zero, we would use our reservoir. As the water from the reservoir enters, that's going to push the air bubble backwards. And we also have a beaker of water to make sure that we don't run out of water in our setup and in our apparatus. So, just like with any experiment, let's talk about our variables. Our first variable is our independent variable. Now, a good example of an independent variable in this investigation could be temperature. And to change the temperature, maybe we would put a heater next to this plant. We could also talk about wind speed, in which case we would use a fan. We could maybe think about humidity, in which case we would kind of add a spray bottle and a plastic bag over the piece of plant to make it very, very humid. Or we could think about the light intensity, and we would change the distance from a lamp or the brightness of the lamp as well. Lots of different independent variables there, and you need to think very carefully about how could they be changed. Our dependent variable, so our independent could be temperature, could be wind speed, could be humidity, or it could be light intensity. Our dependent variable is going to be our rate of transpiration. And that is going to be our distance moved by the air bubble divided by time. Our control variables, well, these are what we need to keep the same. So this is anything else that is going to affect the rate of transpiration. So if we were investigating temperature, we would want to keep the wind speed, the humidity, and the light intensity the same. In addition to that, we want to think about what else is going to affect the rate of transpiration. Well, the size of the plant as well. So the size/species of plant, because different species of plant will have different numbers of stomata. We'd also want to keep the number of leaves the same, because more leaves is going to mean more stomata. And maybe we would want to keep the time that we allow the air bubble to move for the same as well. And then finally, just like with any other experiment, let's have a think about how we can make this setup more precise and more accurate. Well, to increase our precision, we want to reduce the effect of random error. So we're going to take repeat readings and calculate a mean. And then to improve our accuracy, we'd perhaps want to use smaller divisions on the scale. and that means that we would get closer to our true value. So there we have it, everything you need to know about plant transport and the end of Module 2. Make sure you subscribe so you don't miss my videos relating to Module 3 coming really soon.